Welcome again. Today we begin the first of a series of presentations designed to demonstrate how to conduct a practical investigation in biology or environmental systems and societies. While the focus is on the life sciences and on the guidelines for IB students in Group 4, this series of videos would certainly prove useful for all science students. This series focuses on this investigation, looking at the effect of oxygen concentration on the germination and the post-germination growth in Vigna radiata. Vigna radiata are mung beans. Here you can see I have a container of mung beans, and in the container I have some of these tiny packets. They are oxygen absorbers, and they help in the preservation of the seeds and in keeping out small insects, bacteria, and fungi. But in opening one of these containers and looking at this little packet, you might get the idea that you can use these to regulate the concentration of oxygen. Perhaps we can get a closed environment like this. And then we can figure out exactly how much oxygen would one of these tiny absorbers remove from this system. And by adding different numbers of absorbers into the system and sealing it, we could create different environments with different amounts of oxygen. But what would be the purpose of such an investigation? Well, many people ask, what's the best way to sow seeds? Should we place the seed some distance into the soil when we sow them and then add water? Or should we germinate the seeds first by placing them on a moist towel? By placing them in the soil and adding moisture to the soil, you create an environment of warmth and moisture. But in so doing, you may sacrifice the amount of oxygen that the seed is exposed to. Based on these personal observations, asking the question, how much oxygen does Vigna radiata need to successfully germinate, first of all, and then to promote its immediate growth after germination, its post-germination growth? This particular question comes from a personal observation with these oxygen absorbers in this container, and then another personal connection, asking what is the best way to germinate seeds, given the fact that most seeds need warmth, water, and oxygen to grow. So, how much oxygen is enough oxygen? Is it possible that mung beans will germinate with less than 21% oxygen? Or even with 10% oxygen? And what is the optimum amount of oxygen for germination of these mung beans? If I continue to give them more and more oxygen, even beyond the 21% found in the atmosphere, would that continue to increase their rate of germination and their post-germination growth? To do that, I would need a bit more than an oxygen absorber, because the oxygen absorber is simply going to remove oxygen and give me a range of concentrations which are sub 21%. But from another observation that I've made, I know that hydrogen peroxide decomposes naturally to produce water and oxygen. So if hydrogen peroxide of a known concentration is introduced into this container and kept in a separate portion, then in time the hydrogen peroxide would decompose, and in so doing it would raise the concentration inside of this chamber. And using some stoichiometry, I would be able to determine exactly how much oxygen is introduced. So what we propose to do with this experiment is to set up a series of closed containers like these, each with a different concentration of oxygen, going from a very low concentration all the way up to concentrations above 30%. And the goal is to determine at what point will changing oxygen have no effect on the success of germination and on the immediate post-germination growth. In other words, we're trying to find out at what point will oxygen be a limiting factor and when will it stop having an effect or stop being a limiting factor with respect to the success of germination and the immediate post-germination growth. So in addition to outlining what this experiment involves, I've also established a personal connection. Why is it that I would like to know this particular thing? What are my reasons for wanting to investigate this topic? And what thought process did I go through to arrive at my research question and then even my experimental design? In part 2 of this series, we will look at the actual procedure, and I will invite 5 students to assist me in setting up the experiment.